Nature's Archive Podcast, a Jumpstart Nature production.
Have you ever wondered how biologists figure out how plants respond to drought, heat, cold, and other environmental factors? Well, one of the ways they do it is to study phenology. That's a fancy word that accounts for seasonal and cyclical growth patterns of plants and animals. Researchers can look at various attributes, like when a plant flowers, when it leaves out, drops its leaves, and much more. And then they can see how that correlates to other factors like the environment, weather, climate, and even genetics. Today, our guest is Dr. Hanna Makowski, and she tells us all about her research at Black Rock Forest in New York, and surprisingly, why she's had to become proficient with a shotgun to help with her research. Dr. Makowski shares her work on understanding the variations in leaf out and leaf drop phases among trees, the genetic and environmental factors driving these changes, and the broader implications for biodiversity and climate change adaptation. Black Rock Forest is a special place for this kind of research because it's in a transition zone where some southern species and northern species meet. I know that if I ever find myself in New York, I'm going to want to visit. Dr. Makowski's findings could be helpful for tree conservation efforts and predicting forest resilience in the face of global climate shifts. You can find Dr. Makowski on her website at www.hannamakowski.com, on Twitter at Hanna Makowski, and on LinkedIn. And of course, the spelling and links are in the show notes. So without further delay, Dr. Hanna Makowski. Dr. Makowski, thank you so much for joining me today.
Yeah, thank you for having me. I'm excited to be here.
We were just chatting a little bit before I hit record, and this is one of those topics where I know just enough to be dangerous, but I also don't know what I don't know. So I'll probably ask some silly questions as we go, and hopefully you'll bear with me as I struggle through and learn along with what you have to say today.
Yeah, and I'm a postdoc and I'm venturing into a new field as well. So sometimes I feel like I don't know what I don't know, but we'll get through this together.
All right. We're going to talk about some of your research and what you've been working on specific to the Black Rock Forest. But before we jump into that, I think there's some background that will be important to cover. And first of all, I'd like to understand a little bit about you and how you got into the field that you're in. What is the field? And maybe even going way back, how did you get interested in nature in the first place?
So I grew up in a suburb of Chicago, very classic suburbia. We had a little bit of a lawn, but not much exposure to nature. But I was always kind of drawn to flipping over rocks in my backyard, looking for insects, like around, we'd do an Easter egg hunt and I would keep the eggs and put different foods in there and try to keep insects. And I ended up killing them all, but I was running these like mini experiments in my backyard. So I always had this interest in being outside, ripping apart flowers and, grass, and just always felt connected to nature. And then I went on through school, went to college, was interested in biology and thought I was going to be a pre-med, go into medical school. And then just decided med school wasn't for me, as a lot of people do. You have that interest in biology and everybody is like, oh, you should be a doctor. And I did some study abroad experiences and got to see, more of the world and thought like, okay, there's a lot of cool types of plants out there. There's so many different animals, and I'm interested in like what drives this variation that we see. I did a undergrad thesis in floral scent, where I studied the floral scent differences between male and female flowers of this plant down in Florida, Canella winterana. And then I really liked doing research and wanted to keep doing it. So that's how I ended up in grad school. And I went to the University of Virginia and worked with Laura Galloway to study plant mating system evolution.
I have to ask you, the floral scent research that you did, what did you find?
Yeah, so how did we get interested in floral scent in this random plant? There was a grad student who was studying this plant, and he noted it started off in female phase and it switched to male phase. And he was studying the sexual phase transitions and had noted that there were different pollinators visiting the female versus the male phase. So floral scent is used to attract pollinators. And we were wondering if there were differences between the floral scent in the male and the female phase that was potentially driving the differences for the pollinator visitation differences. And we found that the male and female floral scent profiles differed within individuals. So the males and females smelled different within a plant, but there was no consistent male or female scent across individuals, which it's a little speculative, but it's probably good that there's not a consistent male or female scent, because you want pollinators to move between the male and the female individuals of
Right.
different plants and transfer that pollen.
I guess the question I would have, how do you measure the scent?
It's a technique called dynamic headspace extraction. And what you do is it's like a oven bag that people would like put turkeys in or put them in the oven, and it doesn't have any scent to it. So you take an oven bag and you put it around the flower or whatever you're trying to extract a scent from, and you have this little device that pulls air through it, and you capture the compounds in some neutral, like they're these beads. I would have to pull it up from the paper for the exact methods. But basically, you capture the compounds in this medium, and then you can put them into a liquid media, and then you run gas chromatography to identify the chemical compounds.
So yeah, ultimately, okay, you're getting to the chemical compounds that would comprise the scent.
Mm-hmm.
I had this kind of humorous vision of a bunch of grad students all giving subjective
Like
reviews. Yeah.
Yeah.
Which I knew that wouldn't be the case. There's no science in that, but I think it's interesting. We just kind of a peek behind the scenes of how do we get to understanding these relationships that exist between insects and flowers. So thank you for digging back into presumably several years ago research. So anyway, I had interrupted you and you were discussing, I think, how that led you to your next steps.
Right. I really liked research. I liked planning the experiments, and I wanted to keep doing that. And I joined Laura's lab, Laura Galloway, where I was at UVA, University of Virginia. And I came in, had, very little background, and ended up kind of switching away from floral scent. In Laura's lab, I studied plant mating system evolution. I was interested in the geographic distribution that we see of mating systems across species ranges. And I studied the evolution of self-fertilization. And I also studied some morphological aspects of the flowers that allowed them to self-fertilize.
I want to back up because you mentioned that the plants transition from a male phase to a female phase. So is this the same plant, and like for a duration of time, it's producing male flowers and then it switches over to produce female flowers later?
So this is a really cool presentations of sexual phases and sexual systems. Plants can do every and anything in terms of sex phase presentation. So the plant I'm talking about, Campanula americana, each flower is male and then female. So it has a male phase and female phase. The plant that I studied in undergrad, it had also each flower was male and female, but it was coordinated across the whole plant. So all of the flowers were female first, and then all of the flowers were male.
Wow. I have so many questions, but that's probably for another podcast episode.
Yeah. I mean, mating systems there's a reason I studied them for my dissertation. They are really cool. They're cool to study in plants because plants have flowers, which you can manipulate pretty easily. So you can do a lot of different crosses, and like the direct consequence of mating is the transfer of genes into the next generation. So it's a really cool, a lot of biologists study sex, right? Like we're interested in what happens for genes getting into the next generation. So yeah.
Yeah. Yeah. And I admit that a lot of my interests have progressed from non-plants to now, my plant interest is growing. So much of this is new frontiers for me. I think where we're going in this conversation is we're going to talk about phenology, and in particular, some work that you've been doing. But tell me, first of all, like, how would you even define phenology?
Yeah. So can I throw a question back at you to try to do this? Okay. So what are two things that plants, animals, they all have in common.
How high level?
Yeah. Well, maybe so not like cell types or anything like that. Like nothing. I don't want to know what makes the animal kingdom and plant kingdom like similar, but in an organism's life, what are two things that are common between all plants and all animals?
Well, there's definitely a process of maturing that happens over time. And I think they both eventually die. those
Yes.
would be two
Yeah.
things that come to
Yeah.
mind.
So that's what I was getting at. They're born, there's a birth, a seed germinates, an egg hatches, and then there's death, right? And hopefully for that organism, the ideal goal is to reach reproductive maturity, have offspring, and contribute to the next generation. But we typically have a non reproductive, immature, growing stage, and then a reproductive, mature adult. And I mean, there's a ton of variation in what that timeline looks like. Some organisms reproduce once in their life, some live really long times and reproduce a ton, but all organisms go from birth until death. And things happen along the way. And what phenology is the study of those periodic events that happen along the way of biological life cycles, and then understanding how those events are influenced by things like seasonal changes or interannual variations in temperature or even elevation differences. So understanding what time period does an individual become reproductively mature. For me, I'm interested in when trees drop their leaves and when trees grow their leaves again, because there are influences for carbon sequestration. In animals that hibernate, they mostly hibernate in the winter, and flowering happens generally when conditions are good. But yeah, so basically, phenology is the study of this life cycle and how that cycle is influenced by outside factors.
As you were kind of laying out that framework for what phenology is, I was thinking, in some ways, people are simpler than plants because plants, as you point out, at least in mid-latitude regions, they have to deal with freezing and snow and, all these other factors. So they don't just have one pathway of maturing, like they mature, but then there's periods of dormancy and then there's periods of drought reactivity or like other things that could totally change the process they follow. So I can see how this is a fertile area of study to really get into.
Yeah, and it's the reason why I love studying plants. They're stuck where they are, right? So they have to deal with what is thrown at them. They don't have behavioral changes that they can't go avoid drought stress or physically move away to Florida for the winter and go back up in the spring. So they have unique adaptations to deal with the fact that they can't move. And I think that makes them really interesting.
Very often when I think of phenology, I think of in terms of like, when does a plant flower? But as you alluded to, there's a lot of other aspects to phenology. So what are a few of the other things that biologists might look at or measure that relate to plant phenology?
Yeah, so really any part of the growth life cycle. So in a tree, trees put on more bark tissue. So you can measure how much bigger the tree grows, like the diameter over a season. You could also look at the roots. Some people put these bands around the roots and see how they grow over time. Or like, you could dig up roots of something and weigh the dry mass at different points and see how much it's grown. So leaves, leaf out, leaf fall, flowers, basically, yeah, any part of the tree that's growing, you could assess when, where, and how much.
Makes sense. And you were starting to tell me a little bit ago about a dendrometer. And, I think the engineering side of my brain is kind of fascinated by these kind of simple tools. So can you describe for me again how a dendrometer works?
Yeah. So a dendrometer measures tree growth. And the kind I'm using, I'm going to use two kinds. I've deployed one so far. It's called the point dendrometer. And what a point dendrometer does is it has a plunger thing on it that is pressed up against the tree bark. As the tree grows, it pushes the plunger back into the unit, and there's a spring on it. So it measures how far back the plunger moved over a period of time, and that's how much the tree grew. There are band dendrometers, which are exactly what they sound like. They are a band around the tree that as a tree grows, it pushes out. So you can mark where it started and then where it ended.
Kind of like a circumference differential or something.
You measure a tree with at diameter at breast height, and you use DBH tapes, which just account for, you're taking the circumference, but it accounts for pi. So you can get the diameter. And those are good, but a slight fluctuation of where you place the tape, even in the general area, it can change the accuracy. So the band dendrometers are a good way to get a really accurate measure of how much the tree has grown.
Gotcha. And as I think about this, what do we need to understand about leaf phenology or the structure of a leaf or anything else about the leaves to kind of set us up to understand your work? Hey, nature enthusiast, do you want to be part of something bigger? Well, we're building a movement at Jumpstart Nature, and we've just added some new volunteers to help with our podcast and website. But this means our costs are going up too. I need to purchase software licenses to give them access to the production tools we use. For example, one media editing license costs $21 a month. And this is where you come in. Please consider supporting our mission by contributing to Jumpstart Nature through our Patreon or direct contributions, or even purchasing some logo merch. Check out all these options at jumpstartnature.com/donate, also linked in the show notes. Not ready to make a financial contribution? Then please share this episode with 3 friends. Sharing what we do is actually one of the very best ways you can help us. Thank you all for your continued support.
Yeah. So leaves are like the lungs of the tree, right? That is where respiration is happening. And they're also where energy is being created. So trees are large carbon stores. They hold a lot of carbon, and they acquire that carbon. They sequester it through photosynthesis, which happens in the leaves. Trees are always sequestering carbon in their bark and their roots, but they're actively uptaking it when they have leaves on the tree. There is variation in how long leaves are on the tree, and that's sort of what I'm interested in figuring out, what drives that variation, because that variation has implications then for tree growth and carbon sequestration. Yeah, and there's lots of different ways that people measure phenology and think about tree leaves. So there's global scales where you could look at satellite images and see how green an area is to determine when a season starts or a season ends.
So I'm sure everything in biology and ecology, there's just so much variance and there's always a lot of nuance. But in general, can you tell me a bit about what actually triggers a tree to leaf out and maybe drop its leaves later in the year?
Yeah. So I don't know if you've ever gone on one of those websites where you can track fall and find when peak fall is going to be in your area, which is nice if you're planning to go leaf peeping or something like that. And those models take into account day length and temperature. So the amount of light the tree is receiving and then its temperature environment. And they're pretty strong indicators of these large geographic patterns. And people are working to tease them apart because oftentimes, right, they're connected.
Gotcha. And I'm guessing that would, some species, maybe it's the photo period or day length is more important in that. And other species, perhaps temperature is more important. There's probably different combinations for different species.
Yes, that's true. So there are different combinations, and then there is even variation within a species. So I recently came across this cool study that was done in Fremont cottonwood. It was Hillary Cooper, I think she's at Northern Arizona University at the time. They did a common garden experiment where they had individuals from populations of this Fremont cottonwood plant or tree from a latitudinal gradient. They grew them in the south and in the north. So you had populations from the north and the south, and then the same thing up in the north. What they found was that the southern populations seemed to be insensitive to photoperiod. They progressed phenologically with temperature cues, whereas the northern populations went based off of day length. Their interpretation of that was that these northern populations, they've been in an environment where they're subject to harsher frost that could damage them. Temperature can fluctuate a lot and change a lot. Day length is a pretty consistent cue. It changes, but not on a perceptible scale across years or decades even. On the south, where there's not as much frost pressure, having a cue like temperature, if it's warmer for longer, those trees can take advantage of that. Versus in the north, if it's warmer for longer, those trees won't take advantage of that, but they also won't get hit by an unexpected frost.
It's kind of like a built-in risk assessment that the trees have. And so in the south, it's like, all right, the moment it warms up, I'm going to leaf out and take advantage because my risk is low. My risk of a frost and losing all these leaves is low. And in the north, it's kind of the opposite of that.
Exactly. Yeah.
That's really fascinating. And I've heard of some similar sorts of, and I don't know how scientific these studies were, or if it's just more kind of anecdote, but certain plants and shrubs here in where I live in California will have different phenologies based on if they grew, say, on a north slope versus a south facing slope. And now I'm curious and I want to look into this a little bit more because it sort of makes sense.
Yeah, there could be temperature differences or even like the amount of light they receive.
Yeah, definitely. I guess it would probably be both in this case. Very interesting. So even within a single species, there's a lot of variance. And I guess that should not come as a surprise, but not something I think about very frequently. In the Fremont Cottonwood example, you mentioned it was a common garden approach. Can you explain what that is?
A common garden approach is when you take individuals from different locations and you grow them together in a garden, a common garden. And so what you can do typically is you can get individuals from population across some sort of gradient. So this could be elevation, it could be latitude, and you grow them all in a common environment. And oftentimes you have multiple of these common gardens. So if you have high, mid, low elevation, you have a low common garden that has high, mid, and low individuals, and then so on and so forth. And what this does is it allows you to separate out the effects of genetics from the environment, because in those cases, you're holding the environment constant. So you can see the genetic effects of the populations from that high, mid, and low elevation and see how they respond in that common environment. A big guiding equation that we think about when thinking about phenotypes is G by E equals P, and that's genetics by environment equals your phenotype. So you're varying the genetic composition of the individuals in the garden, but you're holding that environment constant. And then that lets you see how G affects the phenotype, the genetics affects the phenotype.
Okay, that makes sense. And it sounds like this is a fairly common approach to studying this.
Yeah, common gardens are common.
Nice.
And they are really powerful.
Can you tell me a little bit about some of the applications of this type of research? Like why would this be important scientifically or otherwise for people to know about?
Yeah, why do we care about when things happen besides, planning our fall leaf peeping trips and fall festivals and that sort of thing? So climates are shifting. We're having earlier springs and later falls. And it's important to know how species are going to respond to these shifts, to know whether, they're going to be able to respond or they're going to have negative effects that impact their ability to persist. And I think this can, there can be impacts that affect the individual's ability to persist. Like, if there's a frost that kills that individual, there's that effect, that individual dies. But then that individual also is not existing in this silo. It has interactions So understanding how these mismatches in, the life cycle timeline, how shifts in seasons are going to impact that, have implications beyond just that individual. And you can have mismatches between pollinators and the timing that the flowers come out. So if, for example, flowers are driven by day length and they don't shift when they're flowering, but you have an insect that is a pollinator, its hatching is driven by temperature. You could have an early spring where that insect comes out way before that flower even flowers, and then that insect doesn't have its food source or home, and then that flower doesn't get its pollinator services. So there can be wide-ranging effects to these mismatches between individuals that experience like the effects of these shifts.
Right. When you think about the system as a whole, there's so many things dependent on each other with, some sort of relationship. And I think the pollinator example is really an important one. And I imagine that there's probably even, say, like annual flowers or seasonal flowers that grow in the understory that need that early open overstory to grow before the trees do leaf out. And there could be some mismatches that occur there with climate change too.
Right. Yeah. Like if the trees leaf out earlier and it's now a dark canopy floor, those flowers might not do so well.
Right. The other thing that came to mind is how weather fluctuates a lot. So having, a season or two where there's a mismatch, things can persist. But if it becomes year after year due to broader climate change, then yeah, that's a different story. Maybe this is a good point to now get into the core of your work. So you're at a place called Black Rock Forest. Can you tell me what Black Rock Forest is? Where is it? What's it like?
Black Rock Forest is a nonprofit organization in the Hudson Valley. It's in Cornwall, New York, which is about an hour north of New York City. And the goal of Black Rock Forest is to promote research, education, and conservation. So we get a lot of school groups that come up. We have a lodge where school groups stay overnight and they go on research visits. The Black Rock Forest is a little under 4,000 acres, so we manage invasive species and conserve the area. The research arm of Black Rock Forest is kind of where I'm at. What's really cool about Black Rock Forest is that as a field station, it's unique in that it's not owned by a single university like most field stations are. It is managed by a consortium of over 17 institutions in New York that make decisions about how the forest is going to move forward. Members from those institutions can come and do research here. We have a lot of visitors, especially throughout the summer, to set up their experiments. More recently, Black Rock has created my position, the postdoc position. I'm the second postdoc that they've had. The goal for them is to start expanding their own in-house research. So that's where I come in.
And then the forest itself, you mentioned it's 4,000 acres. Can you tell me a little bit about what the habitat looks like, what's growing there?
Okay, so Black Rock Forest is, a small forest, but there's a lot of biodiversity. So we have some swampy areas. It's like a mixed eastern hardwood forest. There's a ton of red oaks, chestnut oaks. And what's unique too about this location is that we are kind of at the point where a lot of southern species, we're at the kind of northernmost part of the southern species ranges. And then we're at the southern part of the northern species ranges. So we get an interesting mix of north and south species kind of meeting.
Kind of a transition zone.
A transition zone, yeah.
What specifically are you studying? What are you measuring in the forest?
Yeah, so I am interested in what underlies variation in tree leaf phenology. As I mentioned, right, we know across large landscapes that things like temperature and photoperiod are influencing the start of fall, the start of spring, that sort of thing. But you could be walking through a forest and see two sugar maples side by side, and one has started to turn orange and the other one is still green. or one has totally leafed out and the other one is just starting. And we don't know what drives that variation. And variation at the level of a population or intraspecific variation is important to understand because individual organisms are what is real, right? Populations, species, these are all concepts that we put on to individuals. And individuals are the level at which natural selection acts. Individuals either live or die. So understanding how much variation you have in individuals within a population is important for understanding if that population can persist. Like, do you have enough genetic diversity in a population for it to be able to respond to harsh selective events? Or
so having a lot of variation within a species is sort of a proxy for the resilience of that species?
I think it can be, right? So if you have a population that is all the same and there is something that is negative and so it affects all of those individuals in the same way and they all die, right? That's bad. But if you have 50% of your individuals doing one thing and 50% doing another, if half of them die, the other half can live. So yes, I think it can be a proxy for like ecosystem resilience or that species' ability to persist.
So I'm starting to see how this all connects when you're talking about climate change and other factors that could put a forest at risk. I see the straight line connection now with the research that you're doing. And what is the research though? Like, how are you measuring this?
Yeah, so I have tagged 550 trees across 4 different species of trees, and those are the species I listed earlier. Those are the ones I'm familiar with. I have red oak, chestnut oak, sugar maple, and red maple. And across fall, I was tracking leaf color change and drop. And then in the spring, I've been tracking leaf out. So what that means is I go out and my 550 trees I have tagged, and they're in 3 different loops. So I do a loop a day. So I visit each tree every 3 days. And I have a scale, which I categorize how colorful they are, the percentage of leaves that are still on the tree in the fall, and then the leaf out progression in the spring. And then for each of these trees, I have their location and how big they are and how tall they are. And then I am currently collecting leaf tissue so I can do DNA extractions and have genetic information. And the goal is to determine whether the phenotypic distance I see between individuals. So that's like if one tree is totally lost its leaves and one still has all its leaves, that's like a big distance versus if they're doing the same thing, they don't have a distance. I'm trying to see whether that distance is correlated with genetic distance or if there is an environmental component to this like local microenvironmental variation that is causing the variation that we see.
Do you have to restrict the individuals that you use in the study to like smaller ones that you can have, get better visuals on or better access to, or is it a variety of ages and sizes of trees?
Yeah, so my constricting factors when I was selecting these trees was they had to be on a trail because I tried going off trail and it just took too long. I wasn't going to get the sample size that I needed. So they had to be on a trail.
Very practical restriction. Yeah.
And then I had to be able to reasonably like assess their canopy without spending an hour trying to, visualize because I was seeing a couple hundred trees each day. So I was trying to get trees of generally the same size to control for that. I did measure them, so I have that I can include as like a correlate in my model. But yeah, I was trying to get also trees that were in the canopy. I didn't want to have understory trees in my assessment.
Yeah, I could imagine an understory tree, there'd be another set of variables at play if those were included. Maybe this is off topic, and if you don't want to talk about it, you don't have to, but you mentioned earlier that you had a bruised rib from shooting leaves. Like, what's up with that?
Yeah. The thing about working with trees, right, is that You have to look up at them, so my neck hurts. And then if you want leaf tissue, you can't just snip it off. Some of my trees I have been using a pole cutter, but to get leaf tissue for extractions, I have had to use a method called shotgun sampling, which you use a shotgun to shoot down a branch of the tree so you can get leaves. And I went through training to, learn all the safety protocols. And I have been going around Black Rock Forest for the past 2 weeks with a shotgun in tow and shooting down branches so I can collect leaf tissue. So my shoulder is feeling a little rough, but it will all be worth it once I get the extractions going and get the data back.
Yeah, something that I think the average person wouldn't think about when it comes to being a forest biologist, or what would be the label that you would put on it?
You could call me a forest biologist. On all my job applications on my CV, I call myself an evolutionary ecologist. It's not something you would typically expect. I mean, if you work with trees, you would maybe expect it. An alternative method is to use like a canopy lift, which is possible if your trees are near the road, but I'm deep in the woods. So the shotgun is the simplest way to do that.
And I know that you've been hard at work. It's been field season. So are all of your trees fully leafed out, or you still have measurements you're collecting right now? It's for reference, it's May 20th when we're having this discussion.
Yeah. So all of my trees have fully leafed out. I stopped about a week ago doing my loops, and all my trees were fully leafed out. Some of them hadn't fully expanded to their summer condition yet, but I caught peak variation and I had to prioritize getting the leaf tissue. So I figured it was time to call it on my 8-mile day loops. Yeah, so leaves are fully out and it looks like in fully summer condition. So I do a scale of 0 to 4, and I would say all my trees are a 4 right now.
Gotcha. So it's a work in progress, I know, but do you have any preliminary insights based on the data that you have collected?
Yeah, I do. Definitely from the fall. So I have all that data entered and I've looked at it. I am waiting for genetic data. So I haven't been able to answer my big genetic versus environment question.
I can imagine that will take some time.
Yeah. So in the fall, what was really interesting was, one, there is variation. So that is satisfying. You have these moments where you walk through the woods and you see these things, but when you aren't collecting data, it's all anecdotal until it's in my Excel file. And I think what was interesting was that there is variation within species. And then when looking across species, I see variation in the amount of variation. So my red oaks, they had less variation. They were kind of all more similar to each other and doing things at the same time. Versus my sugar maples, there were trees that were totally green while others had started to lose leaves. So the amount of variation that we're seeing is variable across species. And I think this could be a couple of things. It could be that the red oaks are maybe more photoperiod driven. If they're all doing the same thing at the same time, they could have less genetic variation than the sugar maples. So I am excited to get those sequences and see sort of how much genetic variation is there in all of my species, and does amount of genetic variation correlate with amount of phenotypic variation. And then for the spring, all of my data still needs to be entered, but it looks like spring is shorter. It happened a lot faster and it seemed less variable. So trees were kind of leafing out at similar times. And that would be consistent with what's in the literature. Generally, spring tends to be a little bit more consistent.
It's so interesting to hear you say that, because I'm also thinking about how that tracks, like, say, totally different and probably unrelated, but it's still interesting. And I feel like I need to say it, like bird migration. Spring migration is much more focused and similar periods of time. Fall migration is much more diffuse and spread out and lots of variability in there. And I suppose that tracks with, the plants are, this is just me totally pontificating, but, plants are the foundation of the food web that, say, birds need. So it sort of makes sense if you kind of squint and, think
Yeah.
about it that way. But it's an interesting correlation anyway.
Yeah. And one thing I was wondering too is that I don't know if this connects with the birds, but for the spring, these trees, they're dormant, right? While conditions can vary, based off of their microenvironment. If you're in a wetter area, it might be your roots are colder because there's more water retaining cold, getting to lower temperatures or something. But the trees are dormant. So like springing out of dormancy seems a little bit different than like the winding down. The summer and the fall, you're accumulating so many differences while you're actively growing. Like the effect of the differences in the environment might be stronger at the end of the season than at the start of the season after you've kind of like maybe reset in the winter. I don't know how valid that is, but I like wonder how variable winter conditions are for these trees if they are not actively growing.
I see. Yeah, another level to consider altogether. You had mentioned at one point in time when we were talking about something I wrote down as the leaf economic spectrum. And I wasn't exactly sure where the best place to wedge that into our conversation would be, but maybe now since I brought it up.
Yeah, so the leaf economic spectrum is this global pattern of leaf lifespan and photosynthetic rate. So the economic spectrum idea comes from the fact that a leaf costs carbon to make. The tree has to produce this leaf, it has to grow it. So it has an energy investment that is put into this leaf. And ideally, it will get at least that amount of carbon back, hopefully more, so it can grow. So it needs to net zero or have a benefit. The idea is that you have leaves that have short lifespans. They have to have a high level of respiration. They have to sequester or create carbon, photosynthesize a lot really fast to make up for the energy that it took to make that leaf, versus a leaf that sits around for a really long time. It probably had more investment in, like, if it's a thicker leaf or something like that, but it can respire at a lower rate because it sticks around for longer to make up for that carbon investment that went into the leaf. Something that I'm wondering is I have these early fall trees and I have trees that had their leaves for a really long time. And I'm wondering if this pattern that we see across global scales could also explain what's going on at a local level. So do my early senescing trees have a higher rate of photosynthesis? Did they sort of meet their carbon credits early enough where they could drop their leaves compared to a tree that holds on to its leaves for longer? Maybe it has a lower photosynthetic rate. I don't know if this operates at the level that I'm looking at here in like a population, because we're thinking sometime in the scale of like a week or two. And I don't know if we'll see that, but that is one thing that I am piloting this upcoming summer. I'm going to be looking at my earliest trees versus my latest trees and assessing their photosynthetic rate to see if there's any differences in, the way in which they photosynthesize, the rate at which they do that.
So is then the leaf economic spectrum example that you gave, is that most noticeable like an evergreen tree, for example, compared to a deciduous tree like a live oak versus a red oak or something like that?
Yeah, it's like thousands of plants that they've analyzed, kind of like the length of that they hold on to leaves versus their photosynthetic rate. So I think you'd see that in something that's always green versus something that loses their leaves. Yeah, so I don't know if that scale will hold. I think it's interesting to see if these larger patterns hold true at smaller scales. And I think connecting scales is really important for our understanding of how things work. Like, does what happens at the local level always translate up? And what we see as these broad macro patterns, can we see that on a local level?
Yeah, and I can imagine that in the earlier context of, say, climate change as an example, a couple of weeks difference then amplified by some of these other patterns that may be revealed from your research could really clue you into some other things. As I stumble over my words, but we talked about some of the applications before generally with respect to like understanding how climate might affect some of these trees and which ones might be better suited and things like that. But that could translate into things like evolutionary rescue or assisted migration or other factors that I know you also have an interest in. So maybe you can tell me a little bit about some of these other applications to this research.
Yeah. So I'm working right now in a single population. It's important to understand the context of a species distribution and thinking about your population in the broader range of your species. So in Eastern North America, 20,000 years ago, there was a glaciation. So there's a lot of ice covering the northern part of the eastern US and not a great place for plants to live. So where were plants? Plants were in the south in glacial refugia. And as those glaciers receded, we have expansion from that glacial refugia of seeds, acorns, right? They're making their way more north and they're establishing a new habitat. And now we have what we would consider the modern species range. And with that expansion, you have serial bottlenecks happening. So an individual from a population makes it to a new place and establishes, and then that population expands, and then you get another expansion. And with that expansion, you have a decrease in genetic diversity. So generally, we expect higher genetic diversity in the south of species ranges, at least in the eastern United States, compared to the north. What this means is that populations at the northern range edge, where they're expanding, they have potentially low genetic diversity, which means they may not be able to adapt to changes well. So with climate change, changing suitable habitat for species, what we're seeing in a lot of species is that the south is becoming uninhabitable. Things are getting warmer in the north. The suitable conditions are shifting northwards. And the question remains whether those northern populations are going to be able to respond to this new warm habitat. Will they be able to continue to expand? So evolutionary rescue, assisted migration, these are all concepts moving individuals from higher genetically diverse populations and on the eastern US, that would potentially be the south, and adding those individuals to populations or putting those individuals into new environments where the habitat is becoming more suitable and seeing how that impacts the species' ability to persist and to continue expanding. Yeah, so I think the first step is, I'm doing this at the level of a population, and I have plans to work with collaborators to expand it. And the goal is to understand how genetic diversity is structured across a species range and understand what that means for a species' ability to persist and respond to habitat changes. Because if we find out that low genetic diversity is bad and they can't respond, then maybe we do need to facilitate their expansion or use more southern species to help out the northern populations.
This seems really interesting to me, just simply from the fact that you can't really do an experiment on a timescale of hundreds or thousands of years. You have to really extrapolate here to try to assess how things might play out, and that's I guess what you're doing.
Yeah, and I think I'm a little bit like, I don't know, my research right now is like establishing those natural genetic diversity patterns. I do know there are people who are doing specifically evolutionary rescue experiments. Like the work I'm familiar with is in a guppy population in, I think it's in Brazil. I'd have to read the paper again to check, but it's by Sarah Fitzpatrick at Michigan State. There was this guppy population, and she moved individuals from upstream where, there's a lot of genetic diversity where the native population was, to 2 different sites downstream that had small population sizes, weren't doing too well, didn't have a ton of genetic diversity. And they sequenced all those populations before they added individuals. They knew what they were adding in terms of like the genotypes. And then they tracked what happened after they added these individuals, I think out for like 8 generations or maybe longer. But they wanted to see what happens when you put in new individuals. Do they swamp out the natural population that's there? How do they mix in? Are they like causing harm? Are they bringing in bad, like, is there locally adapted genes there that we're swamping out with the new gene flow? And I think that work is really cool. And when you work with something that reproduces on a shorter time scale than like a tree, I think you can get at some of these questions that can apply to trees. So yeah, I think that there are people who are doing those experiments, and hopefully I'll be characterizing the variation that we do see and alongside of these other experiments. And then I'll be able to like pull from some of those model systems and see, we can combine sort of these different experiments and say, okay, now if we want to do these types of things in trees, if we want to restore a northern population, how should we do it? Let's look at the guppies, or let's look at the fruit flies or something like that as good examples of actually tested evolutionary rescue.
Interesting. Okay, Hanna, this has really been thought-provoking, and there's a lot of things I'd like to follow up on, some threads to pull and look into, and I can only dream that I'll have enough time to investigate all the fascinating things that you mentioned. But, it is wrap-up time. So I always like to ask from a small pool of questions, some standard questions anyway. And one of them is, what's something, that say maybe listeners could do to help prevent or maybe that's blocking them or hindering them from taking action to support biodiversity?
I think I'm going to say too much information, right? There's so much out there. If you're planting a garden, right, there's a bunch of websites for the perfect pollinator garden, or there's a lot of influencers and information on planting local ecotypes and that sort of thing. And I think I've at least seen that can be a little bit debilitating in terms of action. Like if there's too much information, you're like, oh, I don't want to plant the wrong thing, or accidentally create a bee habitat and step on it or mow it and kill the bees if I do the wrong thing, right? I think when it comes to like your own personal garden and your own journey with managing land and promoting biodiversity, I would say maybe don't go plant a bunch of potentially invasive species, but just have a personal goal. Do you want to see a certain type of pollinator? Do you want to attract 10 different types of pollinators, or have pollinators come across a whole season? Like establish a personal goal and then do the best you can to meet that goal, and then learn from it and move on the next year and plant a little bit differently. I think it's like figuring out what your dissertation question is going to be. You can get really lost in all of the information that's out there. And I think when it comes to making personal land management decisions for like your garden, the best thing you could do is start to experiment. We have a fairly new executive director here at Black Rock Forest, Isabel Ashton, and she came from the Park Service, and she managed a ton of national parks out west, and they would do these big experiments, right? And then they'd have to make a big land management decision. And Isabel had presented on her time there, and she said that sometimes you have to make decisions with imperfect data because your experiment hasn't finished running. You don't know the exact trout in that stream that need to be saved. But if you have some data, you can make a decision and then learn from it and move forward. So, I'm coming from this academic background where I need the perfect statistical power to answer my questions. And it's awesome if you want to do your own statistically significant experiments in your yard. But I think just going for it and doing the best you can, noting things down for next year and then trying again. is what I would, what I want to get across. So I just want action.
Yeah. Yeah. Take action.
Yeah. Like, don't blindly take action. Do a little, but there's so much out there. You can't let it stop you from planting your garden. You have to make some decisions.
So I would just mention, I know that longtime listeners already know this, but if someone listening right now is a new listener, we created a podcast episode called The Yard of the Future in the Jumpstart Nature podcast, and had some interviews with Dr. Doug Ptolemy and Leslie Inman from Pollinator Friendly Yards. And I think they do a really good job of trying to make it simple to get started in terms of highlighting some resources where you can find keystone species that occur in your area and starting there. So there are ways to make it simple, but I hear you. There's so many resources, conflicting resources, old resources that are out of date. It can get to be confusing, but I think Doug Ptolemy's Homegrown National Park is a really good place for people to start and find some options for local native plants.
That's awesome. I'm going to be looking up that resource myself. Another thing too, I just kind of want to touch on this concept of natural history. And I think your podcast listeners would consider themselves naturalists. I think natural history can be seen as, same thing with phenology. You're documenting what happens at a certain point. It's cool, but what data are you generating from it? What insights can you gather from just knowing when things happen? This is a great time to be a naturalist because we have tools like iNaturalist, where citizen scientists out there taking pictures of things they just want to identify. But then people who study that organism can use that data to know when something is flowering. And I don't want this to necessarily just be a plug for iNaturalist, but
hey, I plug iNaturalist all the time. The Jumpstart Nature podcast, the other one I do, we just had an episode all about iNaturalist. So I'm all for it.
But even kind of along the lines of your garden too, you can use iNaturalist in your garden. And even if you take a picture of the same plant over time, you could have its progression, and then you can kind of understand its phenology. So even if you just plant one type of new plant and you follow it through a season, there's a whole lot of interest there than just looking at it when it's flowering.
Yeah.
I guess.
The closer you look, the more interesting it becomes, for sure.
Yeah.
All right. So do you have any upcoming projects or anything else you want to highlight before we call it a day?
I mean, I am doing those photosynthesis assays this summer, but next fall I have some collaborators and I'm planning to expand my work and getting people out there surveying fall leaf phenology at the level of the individual. So if you're on the East Coast or anywhere, some of my species go out to the Midwest, and you want to track leaf phenology on trees in your yard or trees at a local park, please reach out. I'm more than happy to talk about collaborating. We can get you, probably not shotgun sampling, but we can figure out a way to get the leaf tissue down, maybe with a pole trimmer and a ladder or something, and include you guys in the broader analysis of the range-wide dynamics.
Well, that sounds interesting for sure. Now, if people do want to reach out to you or just follow your work, how can they do that?
Yeah, so my email here at Blackrock Forest is hmakowski, MAKOWSKI, at blackrockforest.org. I'm on Twitter as Hanna Makowski, and LinkedIn, the same name. I do have a website, www.hannamakowski.com, which has all my research information on it as well.
I will, as always, include links to all of that in the show notes to make it easy for people. So is there anything else that you want to say before we hit the stop button?
No, I think that's
all right. Well, thank you so much for spending a couple hours with me and I guess what is your evening. I really do appreciate it. And I'm looking forward to getting this out to everyone to hear. And I appreciate you. So thank you again.
Thank you.
Thanks for sticking through the entire episode. If you made it this far, I hope that it means that you enjoyed it. If so, please spread the word and share this episode with 3 friends or groups that you think would enjoy it too. As for today's episode, let me know. Did I miss anything? Was there a topic I should have covered? Let me know at podcast@jumpstartnature.com or DM me on any of my social accounts. I'll do my best to answer your questions. You can find me at Nature's Archive, one word, on Instagram, Facebook, and Twitter. I also share photography, nature stories, and much more on those accounts, so you can follow just to stay in touch, too. And despite being called crazy by numerous friends and colleagues, last year I left my tech career behind to start Jumpstart Nature, which Nature's Archive is now part of. For the sake of myself, my family, and the planet, I need to make this work. So please also consider becoming a patron at patreon.com/jumpstartnature. I offer some exclusive content and perks, and you can start donations as low as $4 a month. Lastly, please also check out our latest creation. It's the Jumpstart Nature podcast. We just completed our pilot season, where each episode reveals an unseen, surprising, or misunderstood nature topic with the help of experts and our host, Griff Griffith. It's entertaining and inspiring, and even reached number 3 on the Apple Nature podcast charts. There's much more on our roadmap, but we need your support, so check out jumpstartnature.com for more details. Thank you.